So my research has always been on synapse formation. By always, I mean ever since I started postdoctoral studies, and even to some extent in my thesis. And for many years, and up till now, we've sort of had 2.5 projects aimed at this goal. One is about how when a growth cone finds a potential target, the cellular partners interact with each other to make a synapse. And a synapse, as I'm sure you know, is the site of most of the information processing in the nervous system. And so its differentiation is required for it to be able to process information, for it to transmit information at high frequency, and for it to be able to modify its activity with experience, which is what we assume to be the basis of learning. And we've studied that at the skeletal neuromuscular junction, which is a stupid synapse. It doesn't learn anything, but is very big and very accessible, and pretty much looks and functions like central synapses that we wish we could study and would study if they weren't so small and deeply hidden within the skull. And so essentially that project, which is the longest running one in my laboratory, has involved combining cell biology, molecular biology, mouse genetics, increasingly, in vivo imaging, to find out what it is that the pre- and postsynaptic partners tell each other in order to organize a synapse and get it to a mature state. The second project is about synaptic recognition, that is, going back a step. For synaptic circuits to work, all of the axons have to synapse on the proper postsynaptic cells out of zillions of potential partners, and in many cases on the proper part of the proper postsynaptic cell, the dendrite versus the soma versus the axon, a spine versus a dendritic shaft. And so that is not something the neuromuscular junction is very good at. And so for that, we really do have to take the bull by the horns and go into the central nervous system. And so there we're looking in the visual system. And the particular question there is, retinal ganglion cells are quite similar to each other, but individual subsets of them do various things. So that some retinal ganglion cells will respond when the light goes on, some will detect shadows by responding when the light goes off, some are sensitive to motion, some are sensitive to static objects, some are sensitive to blue things, some are sensitive to red things. And so our project there is to find molecular markers for these subsets and then use them to find out how they make their particular synaptic choices that allow them to form these essentially parallel pathways of information from the eye to the brain. Then going back to the Center for Brain Science, where the overall goal is to look at circuits in a more complicated way, the hope is that as we find out how individual synaptic circuits are developed, we'll come up with ways to visualize them and to manipulate them so that we can see how they contribute to behaviors, in this case visual behaviors. Then the last half project is a full project that my lab does half of, and that is one that is trying to get better ways to see neurons and synapses and to see connections form in living animals. And that is a collaborative project with Jeff Lichtman. And it's combined work in my lab on crafty ways to make transgenic animals that express different colors of fluorescent proteins in different neurons with just brilliant capabilities that Jeff and his colleagues have worked out for imaging fluorescently labeled cells in vivo, and now increasingly computer algorithms for looking at very, very large data sets that this kind of experiment generates and parsing out or segmenting out individual neurons so that circuits can be assembled.